All right, so now we're going to start getting into each of the different isotypes of antibodies. Now, IgM is, of course, always the first, all right? We know that it's the first antibody made. When IgM is secreted from a plasma cell, it's secreted as a pentamer. So you actually have 5 antibody molecules joined together by what we call a J chain, okay? The composition or structure of the antibody molecule other than that is the same. You've got your heavy chains, your light chains, your constant regions, your variable regions. So again, it's the same type of molecule that we've already looked at, but now it's just joined together. So you actually have 5 molecules joined together. Now, a couple of things to think about here. If you have 5 bivalent antibodies joined together, how many antigens could IgM potentially bind? Okay, and you can just go around and count, but it's 10. IgM has the potential to bind 10 antigens. So now here's the tricky question. Are they the same antigens or are they different antigens? All right, hopefully right away you're like, well, that's a dumb question. It's the same. Absolutely it's the same, okay? Remember, you've got 2 heavy chains, 2 light chains. They're identical, okay? It's not going to change because there are 5 molecules. So IgM pentamer binds 10 antigens all at the same specificity, okay? So that's an important point to know for IgM. Now, what are some of the functions of IgM? Obviously, we said it could bind 10 antigens. We call that valence. Okay, it has a valency of 10. We've said that most antibodies are bivalent, so we know that they can at least bind 2 antigens. The primary role of IgM is that it traps free antigen. Okay, so we produce it very well in a primary response when we do have a lot of antigen around, and its primary role is just to trap that antigen and help remove that antigen. Now, another function of IgM is that it activates complement along with another antibody molecule, and I'll give you a second to think about it. There are 2 antibody molecules that activate classic complement, and of course IgM is one and IgG is the other. And remember, the mnemonic or whatever you want to use to remember that is that GM is a classic. So remember that IgG and IgM can activate classic complement. As far as any other functions of IgM, the main thing you have to know about IgM is it's always first. So anytime you get a question on a scenario, and they will confuse you on this, they'll give you a whole scenario about somebody that had a bee sting. They had an anaphylactic reaction. So right away you're thinking IgE. And they're going to ask you what the first antibody made in that reaction was. The first antibody always made to any antigen is IgM. So don't get tripped. Anytime they're asking you about first, it's always going to be IgM. Okay, so keep that in mind. Now, IgM, being so large, we said, had this valency of 10, okay? So we would say that IgM has a high avidity. And all avidity means is the more binding sites, the higher your avidity. So obviously, IgM, of all the antibody molecules, has the highest avidity, all right? Now I'm going to let you think about what another one might be that we're going to talk about in a little bit. Of the next highest avidity would be the one that's a dimer, that's IgA. So you have IgM, IgA, and then IgG, IgE, and IgD. Okay, so avidity corresponds or correlates with the number of binding sites. Okay, now affinity is the interaction only between your antigen and your FAB region. And IgG generally has the highest affinity. Okay? Now, when we talk about somatic hypermutation, affinity maturation, we'll get into why IgG has the highest affinity. Okay? But the interaction between antigen and those variable regions of IgG is a much stronger interaction than you have between the antigen and the variable region of IgM. So, avidity and affinity. IgM is a higher avidity antibody than IgG. IgG is a higher affinity antibody than IgM. Okay? So, affinity and high affinity are going to associate with IgG, and highest avidity, of course, is going to be associated with IgM. We're going to talk now about class switching. So, what has to happen for class switching? You need a T cell, you need CD40 ligand, and you need cytokines. Okay? And now, what we're going to do, and you've seen something similar to this already, now a B cell gets a signal that we're going to switch from IgM, okay, to one of the other antibody isotypes. Okay? So now, when we switch to a different isotype, it's a one-way ticket. We can't go back. We can't do it again. All right? But what you need to notice in the case of the switch is that the variable region doesn't change in the switch. Okay? Now we're really messing with the other constant regions. Okay? Now, when does isotype switching occur? It occurs during the immune response. Where does it occur? Well, it's going to occur in one of those secondary lymphoid tissues. Specifically, where does it occur? It occurs in the germinal center. Okay? So within the germinal center, which is only formed during an immune response, that's where you're going to get class switching. So now we're switching from IgM to one of the other antibody isotypes. The mechanism is similar to the rearrangement process that we've already talked about. You don't need to know specifics of isotype switching. You essentially need to know where it happens and what are the requirements. Okay? But again, you can see we're looping out the DNA, and what we're doing now is we're going to switch to an IgG1. Okay? So IgG1. And all of the intervening constant regions are going to get looped out, and they'll be gone. And the antibody now that we're switching to becomes an IgG molecule. Okay? It occurs in the germinal center. It occurs during an immune response. All right? And remember the requirement for T cells. So this graph differentiates for you between the primary and the secondary antibody responses. I'm sure you've seen this graph while studying immunology. It's a very basic graph, but it really does have a lot of important information. Okay? So as you're looking at the immune response, and it's measured by the antibody titer, so how many antibodies we have over time. Okay? Now, the first time you're exposed to an antigen, all right, the first thing you want to notice is that we don't even have a response for quite some time. All right? We call that the lag period. And in a primary response, it's 1 to 2 weeks it takes for you to even get a decent immune response. Okay? What you finally do, your immune response kicks in, your innate response is working very hard during that 1 to 2 weeks, and now you do start to get a decent antibody response, but predominantly it is IgM. Okay? Now, what will eventually happen, because remember, in your primary immune response, you're now going to have interactions between your B cells and T cells. Let's just use the lymph node for example. Within a lymph node, once you start really getting good interaction, you're going to start to make a germinal center, and that's where we're going to get class switching. So eventually, we're going to switch over and start making some IgG. Okay? That occurs a little bit later during the primary immune response. All right? And then, as you know, plasma cells die off within about 2 weeks. And after the antigen is gone and there's no more plasma cells being made, your antibody response really kind of dies down. Okay? What you're left with, though, is the memory response, which we'll talk about a little bit later. Now, during your re-exposure, or what you would call the secondary challenge, and here's the important point, to the same exact antigen, okay, you have a very different response. This is your secondary antibody response, and you can definitely see that predominantly we're looking at IgG in a secondary response. So your primary versus your secondary response, your primary is predominantly IgM, your secondary is predominantly IgG. Now, let's say at the same time you're exposed to that same antigen, you were exposed to something else, okay? And what you would do, though, is you would have to start this reaction all over again, okay? So you were exposed here to antigen X, you were exposed here to antigen X, you get that really good secondary response. Now, let's say you were also exposed to antigen Y. Well, because the immune response is specific, you have to start the process all over again. Okay, so that's the difference between primary and secondary, and we're going to show a similar graph when we look at the chapter on the memory response.